Good afternoon again. I think that some people will still join us in the coming minutes. Hi, I will introduce myself, and then I will also introduce you to our two key speakers, so Luc and Merdad. Hi, Luc, and hi, Merdad.
Hello, Tarek.
Good afternoon. Hi.
Good afternoon.
Good afternoon. Let me just give you some before introduce you to Luc and Merdad some few rules. You have a chat at the right corner of your screen. You can ask the question you want. We will have at the end of the webinar a Q&A session. Where Luc and Merdad will answer to all your questions. And note that also we will send you a video podcast of the recording of this webinar by mail in the coming days. And so let's start this event by introduce you to Luc. So Luc has joined UCB Pharma in 2008, and he's now heading the solid state and material sciences group. Before his experience in the pharmaceutical industry, he worked also in the food and plastic industry. And also, I will introduce you to Merdad. So he's powder expert at UCB Pharma in the same department as Luc, and his expertise are in single particle and bulk powder characterization. Sorry. So enjoy the webinar, and thank you, Luc and Merdad, again for this presentation. Enjoy.
Thank you, Tariq. First of all, thank you for also giving us the opportunity to present our view, let's say, on drug development. And maybe just before going to the talk itself, good to introduce UCB a bit, because UCB is a big name, let's say, for us in pharma in Belgium. But people outside pharma might think, what is UCB? And people outside of Belgium might think also, what is UCB? So UCB is a Belgian-based company, but it's a multinational, so with active in much countries, a lot of countries, and in fact, we are a global pharma. We are about 8,300 people. So we ourselves, Merdad and myself, are working in pharmaceutical development. And that is where we are based in Braine-l'Alleud. Braine-l'Alleud is based is south of Brussels, about 20 kilometers south of Brussels, just inside the ring. If you see the ring of Brussels before, in the southern tip of the ring. It's quite a big site. It's the main development site of UCB. So about 2,000 people in Braine. The company itself has a revenue of about 5 billion, which is, it's mid-sized pharma, let's say. And the main emphasis, so really develop new medicines, is on neurology and immunology regarding indications. We are active in the small molecule business, which is about, you could say, half of UCB. So we're not going to speak about biological molecules. So some people might be disappointed now, but it's really on small molecules. But the other half of UCB is really working fully on antibodies and biological molecules. Right, now coming to the subject of today, is in fact, we were saying what should we talk about, or what do we want to bring to the people? And then it said that for us it's always a bit striking, because I suppose that also some young consultants are on the talk today, that when we look for people in our teams, and our teams are really solid state material science, really on the physical side of it, we see that a lot of people have a lot of chemical background, analytically or synthetic chemistry. And there you find a lot of people, and of course we want to emphasize also that chemistry is key in drug development. But another point is the physical side of it. And we will today speak about these physical aspects in drug development. It's less common in education. You don't find such as solid state courses in some courses and curricula in education, but not too much. And the same is for material science. Of course, you have specific engineering courses or directions on material science, but overall it's less known. So that was the basis of our talk. Therefore, today emphasis on physics. 2 slides now. One on time, let's say, because if you look at the development of a molecule, from at the left side, its earliest, let's say, discovery of its activity in the body, maybe. So you find maybe an active pharmaceutical ingredient, up to the point that you launch it towards market and then full manufacturing and bring it to the patient. You go through different phases, commonly known to you all, but I guess, but I will just mention them again. You have a preclinical phase where you really only work in animal studies and you really look if it touches the target in animal and in vitro models. Then you go, that's a big step, to the phase 1 human studies with maybe 10, 20, 30 healthy volunteers, and you check mainly toxicology. Second phase, you're really going to see with patients, is it efficacious? Is there an efficacy of the drug, and does it touch the target that I want to touch? And finally, you go to, if possible, thousands of patients where you really have good statistics and confirm efficacy and safety. This can take, as you might know, 10, 15 years easily. So that's a bit the time frame in which pharmaceutical development works and in which we are both active. If you look at it from a material perspective, then we will today have in mind the, let's say, quite classical flow of having a molecule, but in the end, to bring that to the patient, often a classical way is to give them tablets. And in that sense, you need to ramp up processes from having molecules that you can crystallize or make it in a solid form, that build up particles that create a powder that you mix with excipients, and then you make a drug that you bring into the patient, and there it should do what it needs to do, dissolve, get into the blood, and cure the patient. First of all, honor to who deserves the honor. Chemistry is key important. Yeah. So chemistry is there, of course, to make the compounds that we aim for. So therefore, very important. Second point, biochemistry. Everything what happens in the body, very important. So here is the drug should dissolve in your intestines and then get over the gut wall to your blood, past the liver, and then in the end, by the systemic circulation, get into your blood and touch the organs that you want to. A key thing is, also that a molecule in the end almost always needs to dock in a biomolecule that you have in your body, be it a cell membrane, be it an enzyme, be it another protein. And there it needs to change the way your body works, and in that sense, try to cure a disease. A key thing is also very important is the impurities. There might be impurities in your compound, side reactions of chemistry, for instance, or because of storage, some impurities that form. Nitrosamine is now a hot topic. Therefore, also, you need a lot of people that know how to do chromatography to detect impurities. So again, this is important. But we will not talk about that today because we want to stress on physical issues. And the first thing which happens, which is very important for what will follow, is that a concentrated solution, in fact, as such, often is not stable, and it wants to form crystals. This is a fully physical phenomenon. The molecules stay what they are as an identity, but they will turn into a crystal. And first, I want to spend a bit of time on what is a crystal for the ones amongst you that don't know it as such. Crystals are, in fact, a 3D arrangement of molecules, where you see here, this is more giving a 2D, but you should imagine it in the 3 dimensions, where in this case, as an example, it's piracetam. It's a very old molecule from UCB, crystallizes in a very regular way. Yeah? It's fully regarding crystallographic and 3D principles. It's building up a structure. And it does this because in that way, it can optimally pack in order to optimize the interactions. In this case, hydrogen bonding. Yeah. So all the molecules will be perfectly aligned to, in this case, in a 1-1 fashion, but it can be more than 2 molecules that interact each time to optimize hydrogen bonding. So that's the driver. Another thing is also that you see that it gives the impression of being a quite open structure. But in fact, if you look at the molecules from their true volume that they take in 3D, and that's this kind of filled ball module, then you see that it's a very dense structure. Yeah, densely packed, so also gives opportunities to van der Waals interactions, pi stacking. Yeah? So nature likes to make crystals. In the end, under a microscope, it looks like this. So depending on the type of 3D arrangement, you will have different shapes, different habits of crystals. You can have nicely chunk-like crystals that look almost like diamonds, up to more needle-like structures are almost as thin as let's say, it gives a hairy impression. So, which is not so nice. You can imagine to work further with, but some are really crystallizing very nicely. As mentioned already, but here just putting in the single formula I think we have for today, why it wants to form a crystal, certainly from a concentrated solution. It's not because it wants to reduce its entropy, because going into a crystal is reducing entropy. That's creating order, which is not good from a Gibbs free energy point of view, because that will instead increase the free energy. But because of the very good optimized interactions that you have between the molecules, your enthalpy will go down. And that is very beneficial for the system. And in a sense, we are lucky, because making these crystals, you densify the material. So on a little volume space, you have a lot of the active ingredients, which is very easy for handling and transport and for giving it to a patient. He does not need to swallow hundreds of milliliters or so. He can just take a tablet in the end. So you make an API powder that in the end, and Merdat will speak more about that you turn into a compact structure. Still, ideally, patient really likes to take pills, so that's often the end goal of it. Now, if you think about what a drug needs to undergo and needs to survive, you can see here a kind of trajectory overall of what the drug lives. When you make the drug in a reaction in a factory, you can crystallize it. You filter it and dry it, you get a powder that you might store here. Then again, you transport it often to another factory where you make the actual pill or the drug product. Can be by truck, can be by plane, train, whatever. Another company, you make the final drug product, so the capsule of a tablet that will go to the patient. Again, you pack it, you store it for a while. Again, there's a transport to the pharmacy, can be to the doctor, to a hospital. In the pharmacy, the patient can pick it up, or it's being administered in a hospital. And at the patient's house, it can be in the fridge, in the freezer, in a cupboard as such, warm, cold, whatever, along the conditions that are needed. And it's not being put here, but in the patient, then it needs to dissolve again and then do what it needs to do in the patient. So quite some things that it needs to survive. Now, we saw before that we are lucky in a sense because the system, the nature wants to go to a crystal, which is perfect, as we said, because that's then the stablest form, and the crystals will survive all this and arrive at the patient. There is one big complication that is what we fill our days with for a big part, and that is that often molecules do not just make one crystal form. They can make a lot of different crystal forms. And here I'm showing you just one case, which is a UCB case of rhodocotine, which is an active ingredient of Nupro, which is our Parkinson's medicine, which was known since years, since I think 15 or 20 years, when we developed it in its single crystal form. It formed not two beautiful crystals. And you see it had a melting point of 77 degrees and a solubility typically here in ethanol of 500 mg per ml. Yeah. But when the product was already on the market, yeah, then a second form appeared. Despite studies that had been done before, I have to admit that this was before my time, but apart from that, a second form popped up. So rhodocotine suddenly started to crystallize in another arrangement in the crystal. You can see that the molecule had a slightly different type of conformation. And this allowed it then to crystallize in a different manner and in a manner that was more stable. You see it also forms another morphology of crystals. It is more chunk-like, blocky-like. And the melting temperature was 20 degrees higher than the previous one. And because it's more stable also, its solubility goes down because the crystals don't have tendency to go again into solution as the form one did. So this was a big shock, and it is a bigger story than this, but in the end we needed to withdraw the product for some time to the market in order to solve this issue. So it is key to well know as much as possible the different forms that exist for a molecule. And here was mentioned just melting temperature and solubility, but in the end, all kinds of properties, physical properties, are being touched. These are really two different types of crystals. They will have a different behavior when you want to make a tablet, for instance. They will have another part of flow behavior. So really, when you develop a product, you really should know as much as possible the different forms in order to be able to choose the most appropriate one. And this is in fact why typically during a development, we do a polymorph screening to explore the solid form landscape. So we put crystals in a slurry in different solvents at different temperatures. We precipitate by anti-solvent crystals. We put thermal stress, heat, cool. We grind crystals to see if the mechanical stress creates another type of crystal. We evaporate, we cool it down. And this we do by hundreds of experiments sometimes to make sure that at least we know all the crystal forms that can exist. You could argue, or you could imagine, of course, that, and it's being said already in 1965 by Macron, that every compound has theoretically polymorphic forms, and the number of forms that you have is in fact proportional to the time that you spend on looking for them. So as long as you continue to invest in looking for them, you will continue to find forms. So in the end, you should think, when do I stop? That's a key thing. Now, of course, you only want to know the stable forms. That's forms that only exist for 2 seconds or for 2 minutes and then drop back to a stable form. They are not relevant and they will never endanger your development. So you need, in a way, find a compromise between how long will I look for it and what is the risk that I miss a form. And again, in view of the development phases, we typically do the polymorph screening about 3 times. And so we do it in preclinical before going to human. We do some exploration, mainly in solvents that you use in the process. During phase 1, when it survives the phase 1 already, we do a basic polymorphic screen. And then when it proves its efficacy, the drug, and you say, now we go for it. And to make sure we did not miss anything, you go for an extended polymorph screen, which is really hundreds or can be 400 or 500 experiments on milligram scale to make sure that you did not miss a form. So that's the way we approach polymorphism. To make it more complicated, and now I'm approaching the end of my story, let's say, is that you can not only have crystal forms of the pure molecule, different polymorphs, you can also have, with other components, other molecules, other crystal forms. An API can, mixed with excipients, for instance, create a new crystal lattice which incorporates another molecule, which is called co-crystals. It can do this also with solvent, then you call this a solvent, or with water, then you call it a hydrate. But then, for instance, the water really has a specific position in the crystal lattice. It's not just thrown between the molecules. If you have its charge transfer, for instance, an acid that protonates the API, then you speak about a salt. So all these forms that might arise give you, of course, possibility to tune the properties of the crystals. So you could go deliberately to one of these to make a solubility that you want to expose the patient in the right way. You can even keep it amorphous, which per se is not stable, but that way you could expose more. But it's also a threat because you might suddenly, without wanting it, having these crystals appearing, for instance, when it happens with an excipient that is in your tablet. So we need to watch out for this. This is just to show you that the other molecule is really incorporated in the crystal. So it's really taking its place in the 3D structure. Finally, I want to say, and then I conclude, that even when the pill is swallowed by the patient, there you want it to dissolve in the stomach typically, and then the absorption happens in the small intestine. And a thing which happens there is that it's quite of a roller coaster regarding pH. Yeah? Your stomach is very acid, while the rest of the intestine is neutral pH. So also there you can have precipitation effects, and we do investigations also on the solid state after oral administration. Good. So a lot to watch out. Now I'm handing over to you, Mehrdad. Are you taking screen now, or we don't hear you? But
Do you hear me?
yes, we hear you.
Yeah, thank you. Right. So what next beyond the material? So Luke covered the area on the crystals and also the polymorphs, but what about the particles and powders? So the biopharmaceutics, stability and manufacturing of the drug products, we know that it's impacted by the API physical properties. And the last 2 decades, the measurement and the control of the API physical properties has advanced rapidly, whether you control it by changing the crystallization process or your the way that you produce your amorphous APIs. And also the control of solid state is also well understood. So the way that you can do the measurement on the solid state is also very well understood in the field. But what about the impact of the physical properties of the drug product ingredients? So here we are not only talking about API crystals or amorphous particles, but also the excipients we add into the drug product, such as lubricants, fillers, glidants, on the manufacturability of the drug product. So to look at this, we need to look at the typical steps in drug manufacturing. So here is the generic graph showing typical stages that we have. So typically in pharma, we have 2 typical routes. So either we take the dry route, where we need to actually change the physical properties of the powder, so by granulating. So we make the particles larger in order to be able to make a tablet out of it. So here we do dry granulation to make the particles. So we add excipients with API. We granulate it to form larger granules. Then we have to sometimes mill it, or then we need to mix it with the external phase, again, by addition of some lubricants or glidants. And then finally, we make a tablet, and sometimes due to stability or other type of issues we see, then we need to coat the tablets. There's an alternative route as well, where now we go through the wet route, but on the wet route, instead of using a dry granulation, we use wet granulation, where we add a binder to the powder mixture, and then we have to dry it since the granules are wet, so either using fluidized bed dryers or other type of dryers, and then we go again mixing the external phase excipients, and then we make the tablet out of it. But one thing we need to watch out is the performance of the formulation. So these powder mixtures that we have is not only dependent on its individual component physical properties. So we are talking about the API itself and the excipient itself, the physical properties, but also it depends on the process conditions. So at what condition we are processing the powders, as well as the interaction between them. So we are talking the interaction between the excipients and API, etc. So in order to understand that, so we have a lot of complexity in physical characterization, and typically the question is which type of measurement explains the behavior at each stage of processing of these powders. And the question, the answer is we have a large number of characterization techniques. So here I've listed some of them here, but usually the answer to this question is one characterization technique cannot explain the whole behavior. We typically need to do multiple measurements to address one specific area of the impact of the physical properties on the manufacturability. So now going back to the chart that I previously shown, so here I'm just showing different stages going from the powders to the tablet and what are the stages that's involved. So here's the typical or ideal case that we want to have in the drug development processes. So we have the raw material. Then, of course, we store them in a bins. We need to discharge them from the bin, and then we need to feed them, mix them, and make a tablet, and then coating it. But typically, this is not usually what is happening in reality, because the properties of the raw material is not good enough to make tablet by just mixing. So that's why we bring granulation step here, where after feeding, we mix the internal phase together. We form larger granules. Then we dry them if we are doing wet granulation, and then milling it. So in each of these steps, we can see a wide range of parameters or the properties which could affect and some of the issues that we need to watch out. So for the raw material, of course, the size, shape, density, and how easy flowing the powder is, and also the water content is very important for us. On discharge, when we are discharging the powder from the hoppers, so typically cohesive powder or the powder which are sticky, they don't like to discharge and they form like arching or rat holing. We might have a segregation. So if segregation happened, then that means on the final product that you don't have the same amount of API in each tablet, and that fails the content uniformity test. So here is a collection of the parameters or physical properties that we need to watch and measure in order to make sure as the powders goes through this process, we can achieve a really good tablet. So this is typically a typical stage, but what we nowadays in the pharma industry we are pushing the science forward is actually to remove these 3 steps here, because these are quite costly for us, and the idea is to move the development faster, as well as spending less capital or the investment during the drug development. So by doing that, so we need to, what we need to do is to actually engineer the raw material. So in a way, changing, for example, the morphology or the size of the crystals or the structure initially from the raw material. And sometimes this would give us some large granules or large crystals from crystallization, and then we need to bring another milling step in between. So that is the complexity we have and the decisions we have to make during the drug development, as well as what type of properties or physical properties we need to watch out and measure in order to achieve a good quality drug product at the end and to be able to process the powders. So now I've showed the typical parameters or the properties we need to measure, but of course we have a wide range of characterization techniques. So here I'm listed the major or the mostly used categories. So we need to measure the size of the particles. So the shape of the particle is also important, as Luke showed in the slides. So you could either have needle-shaped particles, or you can have more regularly shaped particles. So for needle shape, the processing of the needle shaped particles are very difficult. So that's why for those type of particles or crystals, we have to go through the crystallization route. So hydroscopicity, how much of water uptake we have. The surface measurement either is the surface energy, specific surface area, or the adhesion. The brittleness is how what is the extent of breakage as the particles they go through the processes. The electrostaticity, because we have the organics, so they tend to electrostatically get charged, and that could sometimes cause problem during tableting, for example, measuring the density. Flowability is, of course, one of the biggest issue, because the powder needs to flow through the process. And of course, the compactability and compressibility is also one of the major characterization we do. But sometimes when we go through the characterization, it's not really clear, or we cannot obtain all the links that we want between the material properties and the performance of the powders during the processes. So that's why we need to apply some statistical analysis like PCA, principal component analysis, or finding some correlation and combining different properties or physical properties together to actually link it to the manufacturability. So now I'm just going to talk about some examples on formulation mode of failure. So of course, as the powder goes through, we face the failures, and we need to investigate what is the root cause of that. So here's some of the examples. So on the tablet hardness, the final, how hard the tablets are. So it could be linked to friability, that it can break or not, the tablets, as it goes through the let's say after tableting we have the coating, and you have a tumbling bed where the tablets are tumbled in a drum, and then you apply the coating agent. So during this process the tablets can actually break. You also have capping, where due to the level of hardness that we have, or also the plasticity or elasticity of the particles, you might form a cap, which is not undesirable. Also, on the disintegration, we could sometimes have problem where if we increase the hardness too much, so we know that there is a correlation between the disintegration time and the tablet hardness, and that would be undesirable, because we need to have a specific or a range of values for disintegration time to let the tablet to disintegrate, and then later on absorb and dissolve and absorb to the patient. Powder blend segregation is also an issue. So here is when we put a powder on and generate a heap. So typically we are talking about multi-component mixtures. We have different components in the formulation, and typically the powder tend to segregate, and it's very critical to formulate the powder such that the segregation cannot happen by mixing some cohesive and free-flowing material. Punch sticking also is another issue. So during tableting, if the powder is too sticky, it might stick to the surface of the punch, and then you cannot actually form a good tablet. And finally, powder flow issues is very commonly known in the pharmaceutical, because we are dealing with very cohesive powder. Here's an example of a hopper. And when we have a too cohesive powder in the hoppers, so they form arching and no particles goes out. So typically, we call this one a GMP hammer. So if the process is not designed properly, or the properties are not controlled in a good manner, then we need to have one of the staff actually hitting the side of the hopper with this GMP hammer to break the arch, and then let the particles to flow out. So to go into detail, so blend segregation, what we are talking about. So here is what we have: a binary mixture, for example, which is randomly mixed. So as the pattern segregates, to define a part, so one of the components to go to one side of the volume, and the rest stays on above. And what that causes is when we do the content uniformity test, the first one passes and the second one fails. So it's very critical to be able to measure that. So one of the techniques that we are using is, for example, FT4 pattern rheometer, where you have a impeller rotates and penetrates to the pattern bed and then moves up. So we repeat this test for multiple times, 10 or 20 times, and then we measure the work that it was required for this impeller to move to the pattern bed. So here's, for example, from one of the examples on the formulation. So we have different phases. So we have lubricants in the formulation, different type of lubricants, fillers, and the glidants. And here's our target. So once we have this stability value of 1, that means we have a fully stable formulation. And you can see as we change the components, of course, we get different level of stability. So we have to choose the best one in the process. Also, in terms of the pattern flow issues, so this is one of the examples of the hoppers that I said. So in the hopper, as the pattern flows out, you might form an arch. So there is no particles or powders going out. So you need to disturb the neck here to let the powder going out. Or in some cases, you could have funnel flow or rat holing forming when some of the powder stays close to the walls and they don't come out. So you need to, again, make a mechanical shock in order to remove it. So these are the type of activities we do, but we have a challenge. And the challenge is at the early stage of drug development, we don't have enough API available to run all of these characterizations. Because most of these characterization that I talked about is bulk characterization, and we need a good amount of material at hand in order to analyze it. So what is the solution that in pharma industry nowadays we are trying to adapt is to be able to link the properties at the single particle level. So we characterize a single crystal of the, for example, the API, and to link those properties to the bulk pattern behavior and how these crystals behave when we have it collectively all together. So the solution is, so we are bringing the particle engineering to actually modify the initial crystals. We have the material science who can do the physical characterization on the single particle level. Also, we get help from computational scientists to model the interactions for us. And by combining these and with the knowledge of the process knowledge from drug substance, from the, let's say, crystallization step, and also from the formulators on the drug product, we can then link this length scale between single particle and bulk pattern behavior. To give you an example, so here is is an example of FPI, which is a needle shape, which is not desirable to process it or to manufacture the drug out of it. So what we did is, by bringing the material science and the knowledge of particle engineering and the DS level and also DP, we can manage to change the crystal habit or the shape of the crystals in different forms. So you can see that the shape is changed. And then this has a subsequent impact on the properties. So here we are showing the bulk density. And you can see for different shape, for the same crystal but different shapes, we can have different properties. So the higher this value of bulk density, the better it is for us. So you can see in one case it went from 0.1 to almost about 0.37. And here you can see the shape factors. So here is the aspect ratio, the ratio between the longest dimension and the shortest dimension of the crystal. And you can see here the lower value means they are more needle shaped. So this is the initial crystal that we had. And this is the engineered particle that we have, which the value goes towards 1, which is more like spheres, or they have the same equidimension. Now I pass the floor to Luc.
Thank you, Merat. Indeed, we come to the end of our talk. Just a couple of concluding remarks, in the sense that, we hope that we showed you the relevance of physical sciences for drug development. We are, of course, quite convinced that this is a very rich field. It's anyway strongly contributing to the performance of the drug in the end in the patient. We realize, certainly when we are looking sometimes for people, that it's a less familiar field in chemical and biochemical teaching and curricula. But we also see that a lot can be learned on the job, of course. And that's our concluding message. And thanks to Sandrine for the crystal structures I mentioned. Good. Thank you.
Thank you, Luc. Thank you, Mehrdad, for this brilliant presentation. That's the moment. If you have any questions, you can ask it to Mehrdad and Luc via the questions tab or via the chat, as you want. Take your time. We'll wait some few seconds. Steven is typing. That's the moment. If you have any question, don't hesitate.
We see some familiar names here.
Yeah.
With pleasure, Stephen.
So we have a question from Mathieu. So what's the biggest daily challenge you encounter? So Luc and Mehrdad, I think like in your both jobs.
It's a very good question. That's why we don't readily answer it. One of the typical daily challenges is, first of all, lack of material, as Mehrdad said. This is, for us in solid state, let's say, really, when you're in early in development, you are on milligram scale, and you have to beg people to get 500 milligrams. And also in the material science, for us, it's also very difficult because there you often need a couple of grams, Mehrdad, and that is also very tough. Another thing, but this is in continuous evolution, is to do things risk-based, because you cannot do all. And in a sense, you need to say, we don't know this fully, like the polymorph screen or like the behavior of powder flow or other things. And you need to accept that you need to live with it for now and take the risk that you might hit a wall at a certain moment, and then you have to solve it, but you cannot try to solve everything upfront. But that is a daily balance of what do we do now and what do we postpone, because you cannot do it all. Mehrdad, if you want to.
Yeah, I think apart from the sample quantity on the physical science, but when we are characterizing powders, so most of the time we are not directly measure the exact property which is linked to the process. So interpreting the actual measurement and link it to the performance or the characteristic of the process sometimes is very challenging, and we need to sometimes to spend some time to see what characterization we need to adapt or to carry on in order to address the performance in the processes.
Mm-hmm. That's right. That's not straightforward.
Thank you for your answers. We have another question from Muhammad. So he said that he would like to know more about statistical methods used to correlate different properties.
Yeah.
Well, I can leave that to you, Mehrdad, but it's,
because in my presentation, I showed we have different categories. So let's say we have particle size, particle shape, adhesion. And sometimes it's not very clear how we can link it or whether there's any combined effect. So by running these statistical models like PCR approach, we could get, let's say, the first component or second principal component, and to see the impact of each of these components, whether it's positive or negative for a specific characteristics.
Mm-hmm. And you can do this with a lot of statistical packages.
Yeah.
That's called multivariate analysis, indeed, as you said. Yeah. Mm-hmm.
Thank you, Mehrdad. We have another question from Philip. So, have you seen some progress in the last decades regarding characterization techniques? and what do you still miss to move further with your work?
I think there has been a lot of advancement. I think Philippe himself already knows, but I would say in the last 5 to 10 years, at least from the material science, we are moving away from the traditional way of characterizing. So at the bulk level, of course, there are very new advanced technique like the FT4 powder method I showed, and also the granule drum, for example. So depending on what process we analyze, so we have more option of characterization. But the complexity that also that is bringing, not everyone they are up to date with the new state of the art and how to interpret the results to link it to the performances. But I think the biggest challenge that we have now is not on the bulk level, but actually on the single particle level. There is still a lot of advancement which needs to be done, at least for the pharma industry where we don't have much of material. If I can predict the powder behavior with a single crystal, that rapidly advances the development. It makes the development much shorter, and it's going to save time, and the drug can be delivered to the patients much faster.
Mm-hmm. And we understand that, in a sense, you could say it like that, God knows how to do it, because everything is in the particle, because many of these particles will create the bulk. So if the science would be there and the techniques would be there, we could, but it's in full evolution. Yeah. Philippe, it's not the field that you target, I guess, with your question, but let's say on the real solid state part, where we often want to identify which polymorph, for instance, is in a tablet or is in a bulk. There we really are looking for advances on spectroscopical techniques, because we aim in the end to, without really destroying a tablet, for instance, to know everything what is happening inside the tablet, and there also advances are happening.
Thank you, Luc. Thank you, Mehrdad. And thank you, Philippe, for the question. We have another question from Muhammad. So what microscope you recommend to detect different crystal forms?
But the microscopes we use mainly to detect different crystal really morphologies. When we speak about habit, and their optical microscopy that can do the job already, but to really get a good idea of detailed morphology and surface effects, then a scanning electron microscope is a very nice thing. Yeah? But the pictures that I showed you were for optical microscopy. Yeah? Of course, to detect really different polymorphs, then you need the golden standard there. The gold standard is X-ray diffraction. But of course, that's not a microscope, but there you can really distinguish the different arrangement in the crystal lattices. And that is in the end what counts, because morphology is a secondary property, but to really make sure that you have different polymorphs, you need X-ray diffraction or another spectroscopical technique.
Thank you, Luc.
Stephen.
Yeah, Stephen is asking another question. So is there any computational prediction for crystallization characteristics to predict crystal shape at given? So I think it's clear for you.
Yeah, it is in a sense. There's no foolproof software, but once you have your crystal arrangement of your molecules, you can, in a sense, predict how the different directions in 3D will grow, and this will make, in the end, the morphology of your crystal. So there exists packages for that, and also packages that describe you how these different surfaces, because each time you will have another part of your molecule which is let's say exposed to the outside. You can also predict the properties of this specific facet or a specific face of your crystal. Mehrdad, I don't know if that's.
And there is another more robust technique, but it's very time consuming, which is molecular dynamics. So actually from that you can put the interactions between the molecules and then you simulate it. So, but there's a lot of physics which is still not fully established for those type of activities, but there is a lot of work currently in the literature. So for example, there was a project one project in UK, ADOPT. So they were trying to predict the crystal habit from molecular dynamics. Also, there was another one on the virtual formulation lab, which they use these molecular dynamics in order to, for example, predict the surface energy of the crystals. So, but I think in that specific field, there is still a lot of improvement which needs to be done, but the process is quite slow in order to model it.
Yeah. Right. Thanks.
Thank you, and thank you, Stephen, for the great question. I don't know if somebody has another question. And don't worry, we will send you, so as I told you at the beginning of the webinar, we will send you the video podcast of this live session. And if you have any other question, you can also ask it through LinkedIn event, the web page. And we'll try to see with Luc and Meredith if they can answer. And that's it. I think that we will end it right now. The match will start soon. And I would like to thanks again Luc and Meredith for this brilliant presentation and for the participation to this webinar.
Thank you, Tarek. With pleasure.
Thank you very much. Enjoy your evening and enjoy the match.
You too. All.
You too.
Bye.
Bye-bye.